Hey guys, today I'm going to talk about microbe-human interactions, infectious interactions, both infection and disease. So we'll have at it. It should be a pretty quick one. Again, it's pretty basic stuff, a lot of definitions, nothing too crazy. So without any further ado, let's get started. Alright, so the first thing we need to talk about is the human host. So generally, an infection is seen in three different parts. Contact, so coming in contact with the pathogen. Infection, the pathogen breaking through our protective mechanisms and getting inside of our body and setting up shop. And then disease, and this is where you have a persistent infection that has pretty much taken over. And it's a continuum of sorts. So it's not an all or nothing process. Generally, you can have contact and infection almost simultaneously, depending on where that pathogen's infecting or what big of a wound you have or whatever the case may be. Oops, this stupid box keeps getting in the way. Alright. So body surfaces are constantly exposed to microbes. I mean, we've learned that time and time again from both doing some things in the lab and just by me telling you there are microbes everywhere. They're ubiquitous, and 99% of them are not pathogenic, but there are a few that are. And so it is our body's process to constantly fight off or prevent the pathogenic ones from setting up shop inside of our body. But we're not perfect, and inevitably these pathogens have unique or developed unique ways to kind of get around our immune system and our body's defenses, and eventually that leads to infection. And this is the pathogenic microorganism penetrates the host defenses, enters the tissues, and then multiply. So pathogenic, this is the state that results when the infection damages or disrupts tissues and organs. And infectious disease, the disruption of tissues or organs caused by microbes or their products. So, we all have resident microbiota. We have tons of normal microbiota. These guys are non-pathogenic, and that is because they live within us, but they don't take advantage of us. As soon as they take advantage of us and try to take more than we want to give them, they become pathogens. So cell for cell, microbes on the human body outnumber human cells at least 10 to 1. So there are tons of microbes on our skin, in our digestive tract, in our nose, everywhere, with a few exceptions, being the brain, the reproductive tract, at least in males, especially the testes, the eye, and the lower lungs. But other than that, you can find microbes usually on a fairly regular basis. So metagenomics, this is the ability for us to do deep sequencing of all the DNA in our body, both humans and microbes, and then taking that information and searching known databases and trying to figure out what microbes are present in our body. And we've discovered that it's not as consistent as we thought it would be. So certain people are infected with certain microbes, and that's what makes their immune system slightly different, or makes them slightly different. So this is like a whole new hot button topic in science right now. People are loving this metagenomics and the Human Microbiome Project and all this good stuff. It's pretty cool. And how do we acquire resident microbiota? So we get these from the environment generally. We can also get them from our mothers and occasionally from our fathers. So normal microbiota you can see pretty much everywhere. Skin, adjacent mucous membranes, respiratory tract, gastrointestinal tract, outer opening urethra, external genitalia, vagina, external air canal, external eyelids, and so on. So they're normally present in and on the body. Human cells in an adult is about 10 trillion. Bacteria is about 100 trillion, so there's a tenfold increase, so there's a lot of microbiota. Primarily found externally, both on the skin and eyes. They're everywhere. Internally, nose, mouth, intestinal tract, vagina, and urethra. Especially the intestinal tract. That's where the majority of microbiota, at least internally, are located. So resident microbes, each site has particular populations and it changes over time, and this is even different between person and person. Transient microbes, so these do not normally reside just passing through. Most are harmless, some are pathogens. So pathogens do fit into this category. So all pathogens are transient microbes, but not all transient microbes are pathogens. Some transient microbes are things found in like Activa yogurt. There are microbes found in that yogurt. They're not normally found in our gut, but when we take in that yogurt, we bring them in, they set up shop. And 9 times out of 10, they don't set up shop for long and they're just passed out through your stool. And then we have the opportunists. So these are all where the pathogens fit in. All pathogens are opportunists and all opportunists are pathogens. That makes sense. They cause disease when given the opportunity, breakdown in immunity, certain medical treatments, and implantations of devices. So these are the guys that like to set up shop when your immune system's weakened. A lot of molds, fungi fit into this category. Excuse me. Molds, fungi fit in this category. They're ubiquitously found in the environment. If you have a healthy immune system, you're fine. Until something goes wrong with your immune system, then they can kind of take over. So how do we acquire these guys? Through the birth canal, from food, from breathing, natural succession, just from touching things in our environment. That's where we get our lactobacilli, coliforms, and our anaerobes from. A lot of these guys hang out in soil. So playing in the soil, coming near the soil, touching the soil, all that kind of stuff allows for the natural succession of these. So impact of normal flora. So biological success for the bacteria. So bacteria like being in our gut. It's very nutrient rich. It's enjoyable to them. They get all the vitamins and nutrients and materials and stuff they require. It's also beneficial for us to have normal flora so that we can exclude potential pathogens. If we have a lot of microbiota using up those nutrients that are available, it prevents pathogens from getting a hold of them. So it's a kind of a competition. And this competition makes it hard for them or for pathogens to set up shop. But also, occasionally, they can provide some nutrients to the host. They can break down things that are hard for our digestive tract to break down and make them smaller and easier to assimilate. They can also improve host nutrition. Vitamin B12 is produced from bacteria. It's a byproduct that they produce, and it's very useful for us. Immune stimulation. So our immune system does recognize these guys as pathogenic, and generally they treat them less severely, or they don't harm them as much as they would a normal pathogen. But it does kind of awaken your immune system and make them realize, hey, these are bacteria, we need to recognize that, and we need to be prepared to attack bacteria in the future. And also have potential negative impacts, especially in people who are immunocompromised or going through some sort of cancer therapy or such. It can be, some of these guys have shown, especially people who have AIDS, to have the ability of transforming themselves into pathogenic microbes once the immune system has left, primarily by plowing through the intestinal membranes and getting into the blood. So these are sites that are thought to be sterile. You can see heart, liver, kidneys, brain, muscle, bone, but I can guarantee you that the good majority of these aren't actually sterile. You'll find some if you really look. Generally, the ones that absolutely need to be sterile are the brain and spinal cord. You'll rarely find anything in there, and if you do, you're in bad shape. Along with the ovaries and testes. Again, you shouldn't find anything in there. If you do, you're in bad shape, especially when it comes to sterility. Blood, urine, cerebral spinal fluid, these all should be sterile, but again, urine tends to have bacteria in it. It's not necessarily a big deal. And occasionally you'll see some things in blood in like a transient kind of manner, and that's not necessarily a bad thing. So, progress of an infection. So, a pathogen, this is a microbes whose relationship with its host is parasitic and results in infection and disease, and generally a breakdown of tissues. And that breakdown of tissues releases more nutrients, and the pathogen sucks that up. Type and severity of infection depends on pathogenicity of the organism and the condition of its host. So the pathogenicity is how good of a pathogen that microbe is and what it is doing to become that good of a pathogen. Now, the pathogen can kind of change its things or its virulence factors to make it more pathogenic, but it also has a lot to do with the hosts that they are infecting. Generally, there are a group of hosts that are easier to infect, and for that reason you'll see more pathogenic infections in them. Primarily in the old age and extreme young, or extreme youth, or the young group. These are people probably under the age of 7. I'd say anyone over the age of 70 is in the old age group. And this is primarily because as you get older, or if you're very young, your immune system isn't fully developed. If you're older, your immune system is kind of breaking down, so you generally have a weaker immune system. Genetic defects in immunity, there are a lot of them, a lot more than I thought until I got into immunology. Not very common, but there are a lot of them. Things like bubble boy syndrome, where you have people who live in a bubble because they have no effective immune system and anything that they come into contact with could be potentially pathogenic. Or you can acquire defects in the immune system such as AIDS, which is the disorder associated with having prolonged HIV infection. Surgery and organ transplant. So anytime you get surgery or organ transplant, you generally go on some kind of immunosuppressive therapy so that your immune system doesn't react to the new organ. But that immunosuppressive therapy does affect your entire immune system, making you more susceptible to other infections. Underlying diseases, cancer, liver malfunction, diabetes, all of these things have effects on your immune system. Chemotherapy, immunosuppressive drugs, again, can weaken your immune system for obvious reasons. They're called immunosuppressive drugs. And then physical and mental stress. So being physically and mentally stressed has been scientifically proven to cause a decrease in your immune system's function. Definitions of terms. So pathogen, this is a microbe that can cause disease in a susceptible individual. Virulence, this is the degree of the ability of a microbe to cause disease in another organism. So the more virulence you have, the more effective you are as a pathogen. Infection, colonization of an organism by another microorganism, or by a microorganism, with or without disease. So I am infected with my normal microbiota. Doesn't necessarily cause disease, but they are setting up shop inside of me. A disease is when it actually causes harm to the host and starts breaking down tissues. Communicable, this is the ability of a disease-causing organism to be spread between individuals. So this is 99% of our pathogenic diseases. They are transmitted communicably. I cough on you, you get it, that kind of thing. And virulence factors, these are the microbial components that contribute to the ability to cause disease in a susceptible host. So these can be things like toxins, really nice fimbriae that attach and make them stickier so that they can stick all over the body, things like that. Things that the microbe has the ability to change to make them more pathogenic. So pathogenicity, so you have two different types. True pathogens, these are guys that only live as pathogens. They need to infect an organism to survive. And opportunistic pathogens live in the environment, can also live on you or in you, but as soon as your immune system is weakened, that's when they become pathogenic. So virulence is the degree of pathogenicity, determined by its ability to establish itself in the host and cause damage. So the key to being a really good microbe is to cause enough damage to where you can get a lot of extra nutrients released, but not too much damage to where you kill your host. And there's always this kind of fine line you have to dance around. And that's what adjusting your virulence factors allows you to do. And your virulence factors are characteristics or structures of the microbe that contributes to its virulence. So it's easy to remember since it's got virulence in the name. Different healthy individuals have widely varying responses to some microorganisms, and that's because hosts evolve. Also because we all have slightly different immune systems that respond slightly different to different virulence factors. So the very first bit about becoming infected with a pathogen is establishing or getting into the body. and that is through a portal of entry. So we have our skin. It's covered with 7 to 10 dead layers of skin cells that have been keratinized. So it's fairly tough, it's fairly robust. So it makes it pretty difficult of a barrier for microbes to get through. But it's not perfect, and we also have portals of entry that are just naturally occurring in our skin, like our mouth, our eyes, our anus, our urogenital tract, and that kind of thing. Because we need to be able to excrete things out of our body, we need to be able to communicate, and for that reason we need holes in our skin. And these all become somewhat susceptible to an infection. Our body has developed ways to get around this by producing things like mucus, or having mucous membranes, or having antibodies being constantly secreted to kind of ward off infection, or ward off these guys from getting in. But there are still ways for pathogens to get around these things. So infectious agents that enter the skin, these are generally through nicks, abrasions, and punctures. Intact skin is very tough, like I said. There are only a few pathogens that I can think of that actively burrow through the skin to get through it. And they are not bacterial, they are not viral. They are parasitic in origin. And we'll talk about those probably in 2 or 3 weeks. They're pretty cool. And then some create their own pathogen or passageways using digestive enzymes or bites. Gastrointestinal tract is a portal, so pathogens contained in food, drink, and other ingested substances. So these are usually the guys that are transmitted via the fecal-oral route, and we'll spend a whole lecture just on these guys. And these adapt to surviving digestive enzymes and pH changes. You can see a whole list of guys that have the ability of doing this. Salmonella, Shigella, Vibrio, all that good stuff. We'll talk about these in more detail later on. Respiratory tract. So these go through the respiratory tract, obviously. Portal for the greatest number of pathogens, and that's because it is okay, so I'm back. Sorry about that. So we'll continue where we left off. So respiratory portal of entry. So you get the greatest number of pathogens entering this way, and that's because the lungs are fairly large when you look at them at a surface area, from a surface area's perspective. There's a lot of surface area within the lungs, and that's important for absorbing as much oxygen as possible, but with that, it allows great areas for pathogens to get in. Also, the skin in the lungs are very, very thin, or the tissues in the lungs are very, very thin, and that is to allow oxygen to assimilate. But it also allows for an easy entry point for a lot of pathogens. You can see a great number of guys down here. We'll talk about these in more detail, but whooping cough, influenza, measles, things that you've probably heard of before and associate with the respiratory tract. Urogenital portal of entry, so this is a perfect place to enter if you're a pathogen, primarily because organisms need to have sex to survive, so why not manipulate that? They also have a very good ability to cause asymptomatic infections, and that is where you're infected but you show no symptoms. And if you're an STD, that is the perfect situation to be in, and that's because I wouldn't want to have sex with someone who was oozing stuff out of their vagina, for instance. Instead, I'd have sex with someone who looked normal, but then I would get infected because they were actually infected but not showing any symptoms. And these guys have a remarkable ability for being able to do that. They enter via the skin, mucous membrane of the penis, external genitalia, vagina, cervix, and urethra, and so forth. All of these areas have usually thinner skin, making it easier for things to penetrate through. They can also penetrate unbroken skin or unbroken surfaces, but generally some microtrauma does need to occur for the guys to get in. Some examples of these: syphilis, genital warts, chlamydia, herpes, gonorrhea, the list goes on. There's a lot of them. You can see the sexually transmitted diseases happen on a fairly regular basis throughout the United States. HPV is by far the most common, but you have trichomonas, chlamydiosis, herpes, gonorrhea, hepatitis B, AIDS, syphilis, all pretty high up there on the list. Pathogens that infect during pregnancy, so this can be somewhat problematic. A lot of these pathogens aren't that serious in a healthy individual who's not pregnant, but as soon as you become pregnant, your child becomes a target. A good acronym to remember these is TORCH. So toxoplasmosis, other diseases, rubella, cytomegalovirus, and herpes simplex virus. Toxoplasmosis, rubella, cytomegalovirus, and herpes simplex virus are all very, very, very mild, especially in healthy adults, but as soon as you become pregnant, it becomes serious for your child. Generally, you need to become infected with these guys for your very first time while you're pregnant at a very specific time in the pregnancy for it to become serious. That's why you don't really hear of it that often, because it is a fairly hard thing to do. These guys have the ability of crossing the placenta. So in general, mother's blood is separated from baby's blood by the placenta. And these bacterial cells, in this case, or some viruses, or even some parasites, have the ability of kind of breaking through the placenta and getting into fetal blood. That's when the problem occurs. So size of inoculum has a lot to do with how serious an infection is, at least in the initial parts of the infection. Some microbes need just a few individual bacterial cells to cause a severe infection. Others need a lot. It just depends on the microbe, depends on how they infect, depends on their virulence factors, depends on a whole bunch of stuff. But for most agents, infection only proceeds if the infectious dose is present. So that is the minimum amount of the infectious agent to cause a disease. And microorganisms with smaller IDs have greater virulence and are more pathogenic. That just makes sense. So once you get in, once you find a portal of entry, you now have to kind of set up shop. And to do that, you need to attach. So to attach, we've talked about this before, you can use things like fimbriae, capsules, or spikes. This allows you to attach to the surfaces you want to be, and you can just sit there and sop up food. Here's a quick list of some guys and how they attach. You can look at that at your leisure. Now you have to establish your infection, and this is surviving the host defenses, or step 3 of our infection story. So the very first thing you'll encounter, at least from the host immune system, would be phagocytes. So these are the vast majority of white blood cells, and their primary goal is just to gobble up, destroy pathogens, along with any other kind of debris that makes its way into the body. To combat this, pathogens have come up with things like these anti-phagocytic factors. Leukocytins, so these are toxic to white blood cells. So obviously if you kill the white blood cells, they can't phagocytose you. That works out well. External surface layer, so your slime layers, your capsules, your biofilms, makes it very, very, very hard to phagocytose because you can become too slippery. And once you're slippery, the bacteria or the phagocytes can't grab onto you enough to pull you internally in. And then some don't even care if they get phagocytosed, they just survive inside the phagocyte. Things like Legionella, Mycobacterium, and many rickettsial organisms. So tuberculosis is a prime example of this. It becomes phagocytosed by macrophages, which are phagocytes in the blood supply. And here they've stopped the fusion of special lysozymes in the macrophage that would normally digest them. So by stopping this fusion, they stop the digestion process, and then they just kind of hang out inside of the phagocyte. So how do they cause disease? Use your virulence factors to break down tissues. By breaking down tissues, more nutrients are released and you get more goodies. So exoenzymes are a good example of this. This is where you'll have your bacteria releasing enzymes that degrade things like plasma membrane or cell walls, and this allows for the bacteria to kind of kill the cell, take up its nutrients. Toxins just shut the cell down. Once the cell shuts down, it releases its goodies. And then you can also block phagocytic responses. So extracellular enzymes, they break down and inflict damage on tissues or dissolve the host defense barriers. So mucinase breaks down mucus. So by breaking down mucus, It stops the mucus from doing its job, which is to kind of slow the microbe down. Keratinase, this breaks down keratin. You break down keratin, you get through the skin. Coagulase breaks down collagen. Collagen is found as a good connector between cells. And once you break that down, that connection breaks and those cells become easy to access. And hyaluronidase is hyaluronic acid is a acid motif found in almost all cells' plasma membranes. As soon as you break that down, you break down the plasma membrane and you can take in whatever that cell has inside. And then some react with components of blood, so coagulase is a good example of this. This actually causes blood to coagulate. And kinases, these break down kinetic activities within the blood. So bacterial toxins, these are specific chemicals produced that is poisonous to other organisms. These aid in the killing of the host cells. By killing the cells release their goodies, the bacteria get to take in all the goodies. So toxogenicity, this is how powerful a toxin is. Toxinosis, a variety of diseases caused by toxogenicity. Tetanus is a good example of this. The disease is not caused by the microorganism directly, instead it's caused by a toxin the microorganism releases. Toxemia, so toxosis in which the toxin is spread by the blood from the site of infection, so tetanus and diphtheria is another example of these. Once it gets into the blood, it's downhill from there. Your blood just kind of spreads it all over and then it can cause systemic death. And intoxication, so this is toxoplasmosis caused by ingestion of toxins. Botulism is a good example of this. There are also a few foodborne illnesses that are caused this way. So endotoxins and exotoxins. So exotoxins are actively released by the cell, and these are used to break down materials within the host. Endotoxins, on the other hand, are toxins that are only released after the bacteria dies. So they're bits and pieces found within the bacteria, and then as soon as the bacteria dies, those bits and pieces get released and cause a toxic effect in the cell. So they both have slightly different functions. Exotoxins are used for actively dividing bacteria. They're used to kind of help a bacteria get its nutrients. Endotoxins are primarily used to aid your friends, because as soon as you die, you're obviously not going to be taking up any of the goodies that you're causing to be released. Instead, you want those goodies to be released so that your neighbors have a better chance of surviving. So exotoxins actively released. Endotoxins are found usually within the cell wall or inside of the bacteria, and they're not released until the cell dies. So this is a little bit about an AB toxin. We'll talk in more detail on what an AB toxin is, but an AB toxin basically has two units. The B unit is what allows it to enter the cell. It kind of sends a trigger in the cell to say, hey, take me in, internal. And then the A unit breaks off and then causes some kind of toxic damage to the cell. And we'll talk about this in more detail later. So the process of infection and establishing disease. So establishment, spread, and pathogenic effects. Microbes eventually settle on a particular target and organ and continue to cause damage at that site. So microbes usually have a preferred place they like to be. Once they get into that place, they settle. That's where they set up shop. They start releasing their toxins. They start releasing their exoenzymes. They start breaking down tissues so they can get their nutrients. This frequently weakens host tissues, which can be, again, problematic. You don't want your tissues breaking down. That's what they're there for. They're meant to be intact, meant to be strong. As soon as they get broken down, bad things happen, such as necrosis. And this is the accumulated damage of cell tissues that can eventually lead to tissue death. So establishment and localization of infection. So the most simplest infection would be a localized infection, and that's where it just stays right in the area of entry, so right at the portal of entry. These are things like pimples, boils, generally more mild, but there are some severe ones. Things like MRSA, for instance, can be a localized infection but can spread, and that's when it becomes problematic. A systemic infection, these can be a little bit more serious. and that's where you get things migrating into the blood, and then they can kind of be transferred throughout the body. Focal infection, that's where you have something that was localized at one point, but then it transfers itself to the systemic region, usually by co-infection. Mixed infection, this is where you have many different microbes in one area. And then primary and secondary infection. So primary infection is where you get infected with something, and that weakens the immune system in that area, which allows for a secondary opportunistic infection to occur that normally would not occur. So now we'll talk about a little bit about causing disease, but this is basically the 4 steps, or the 4 steps that we've gotten to so far, but the 5 steps total of what a disease is. So it's finding a portal of entry, so breaking through the skin, getting through the GI tract, getting through the respiratory tract, and so forth. Then you're attaching firmly to the area that you want to set up shop in. Then you survive the host defenses, combat the immune system, stop phagocytosis, protect yourself. Then you start releasing your toxins, you cause damage, you break down tissues. By breaking down tissues, you get more nutrients and you survive, you reproduce. Once you reproduce in significant numbers, then you can start exiting the host. So if you don't exit the host, then as soon as your organism of choice that you're infecting dies, so do you. So you need to have the ability to kind of release yourself into the environment so that you can spread. So the progress of infection. So this is this little bit here, the causing disease and damage. So once you get through all of this stuff, this is when you actually cause the disease. And these are the different stages of disease, at least from our perspective. So the first bit is the incubation period. The incubation period is where you have fairly low numbers of microorganisms present, usually not enough to cause that many symptoms. It's kind of like the getting stronger period. It's where the microorganisms are dividing, they're getting used to their environment, they're kind of setting up shop. Next you have your prodomal stage. So your prodomal stage is where you have a critical number of bacteria that can cause pathogenicity, but it's not the number needed to cause the actual infection or the symptoms associated with that infection. Instead, you get some symptoms, but they're different symptoms. So this is generally seen as kind of like a malaise or a sleepiness or being tired, or that general ickiness feeling you feel like when you're about to become sick. That's the prodromal phase or stage. It's where the pathogen is beginning to set up shop, it's beginning to become pathogenic and cause disease. But the symptoms aren't the symptoms associated with that disease normally. Then you get into this period of invasion where you get a heightened amount of symptoms, and these are the symptoms that are associated with that disease. So if we're talking about a cold in this example, your incubation period you feel no symptoms at all because you only have a few virus particles in your respiratory tract. Those virus particles then divide, or infect, become many more viral particles. Those viral particles then start to cause some symptoms, but these symptoms aren't generally associated with a cold. It would be things like getting tired or being sleepy or just generally feeling icky. Then, during this period, you have more and more viruses. Those more and more viruses can infect more cells. That's when you get the actual symptoms of the cold, so you get the runny nose, the headache, all that good stuff. And that's when you get the height of your infection. Then your immune system kind of takes over. Your immune system starts to fight off the infection. And then as the infection gets weaker and weaker, thanks to the immune system, the symptoms get less severe, and this is called the convalescent period, until you fully recover. Now there are always people that are going to be chronic carriers, and these are people who always have a small number of microbes in their body, usually too small to actually cause any of these symptoms, but they're always present, and for that reason they can be transmitted. So a sign is any objective evidence of a disease as noted by an observer. So a sign is something that can be physically seen by an observer. So you look green, you have jaundice, you're heart rate has gone up, that kind of things, things that can be measured by a physician. A symptom, on the other hand, is the subject evidence of a disease as sensed by the patient. So a symptom would be things like, oh, I have a sore throat that can't be seen, or I feel achy. That can't be objectively seen by a doctor. It needs to just be described by the patient. And then a syndrome, this is when a disease can be identified or defined by a certain complex of signs and symptoms. This is what makes a differential diagnosis work. So by saying a cold has this, and this sign, this symptom, and so forth, we can say, okay, if anyone gets those signs and symptoms, they probably have a cold. So here are some signs. You can see fever, septicemia, skin eruptions, leukocytosis. All these things can be measured by a physician. Then you have your symptoms, so chills, your pains, your malaise, your fatigue, chest tightness, itching. All of these cannot be measured by a physician. You just have to go on the patient's word. Signs and symptoms of inflammation. So you get fever, pain, soreness, swelling, edema, or buildup of fluids. Granulomas and abscences. So granulomas are where you get white blood cells kind of walling off an infection, and they cause these nice little kind of lumps of white blood cells. And if they get large enough, you can get something like an abscess, which is just kind of a huge lump of white blood cells. Lymphadenitis, this is where you get a swollen lymph node. This is primarily because the lymph nodes are used for collecting and draining activated immune cells, and for that reason they get larger during an infection. And then lesions, this is the site of infection or disease. Alright guys, so we'll continue on where I left off. Sorry about the breakup, having more technical issues. So we'll continue on with signs and symptoms and how they present in different parts of the body and so forth. So I think I've already talked about this slide. Signs and symptoms of inflammation. So generally fever, pain, soreness, swelling, all of these things are systemic effects that are associated with inflammation. Edema as a hallmark of inflammation. This is a buildup of fluids. Generally that's what we consider inflammation to begin with. Granulomas and abscences, so these are again just conglomerations of immune cells that kind of wall off infectious things or non-self particles, and they can form these nice little like nodules. Lymphadenitis is swollen lymph nodes just from the draining of lymphatic tissues and cells into the lymph nodes. And then lesions, this tends to happen at the site of infection or disease. You get stuck in the arm with something like E. coli or some kind of nasty pathogen, you'll get a nice little kind of swelling that occurs in that area. And you'll tend to break down tissue in that area because that's where they're the most concentrated, at least initially, and that's when you'll get a lesion. Signs of infections in the blood, so you'll get change in number of circulating white blood cells. It's either leukocytosis, which is an increase in the number of white blood cells, or leukopenia, which is a decrease in the number of white blood cells. Depending on what the cells infect and what they do to the body, that determines what type of change in the white blood cells will occur. Leukopenia is most commonly found in things like HIV. That's because they target white blood cells. But leukocytosis you'll find in a lot of bacterial infections because you're increasing the number of white blood cells to ward off infection. So septicemia, this is a general state where microorganisms are multiplying in the blood are present in large numbers. So septicemia is where you actually have microorganisms in the blood and they're multiplying. So this is very severe. If you have microorganisms multiplying in the blood, you are in serious trouble. And that is because they can pretty much get as much nutrients as they want from the blood, and it allows them to travel throughout the body fairly easily. Bacteremia or viremia, so these are microbes that are present in the blood but are not necessarily multiplying, so it's kind of a transitional state. Septicemia is much more severe than bacteremia. So portal of exit, like I've mentioned before, this is very important in continuing the life cycle of a pathogen. Here are some of the more common portals of exit. Coughing and sneezing, that's an obvious one. Cells or skin cells or open lesions, not as common, but it is present. Urine, especially in sexually transmitted diseases. Feces in the fecal-oral route. removal of blood and a lot of bloodborne pathogens, and insect bites. Insect bites are also very important, especially for tropical diseases like malaria. So exoportals. So most commonly it's the respiratory and salivary glands, and that's because we're constantly releasing saliva, we're constantly opening our mouth, we're constantly coughing, we're constantly sneezing. So it's an easy way to get out. Skin scales, not nearly as common, but there are some organisms that prefer to live right below the skin surface, so it's easy for them to kind of slough off with the skin scales. Fecal exit, this is very common in the fecal-oral route, so a lot of your gastrointestinal disorders are transmitted this way. Your genital tract, all of your STIs are transmitted this way. And then removal of blood or bleeding, so bloodborne pathogens fit in this category. So microbes, if you're a good pathogen, you need, like I mentioned before, you need to be able to persist in your host of choice without necessarily killing your host. And one way of doing this is by becoming latent. And latent is where you kind of have a period of time where you're not really infecting or causing damage to the host. Instead, you're just kind of hanging out, low numbers, waiting for a good opportunity to arise for you to kind of wake up and take over the body again. A lot of viruses are very good at this, so herpes simplex 1 and 2, hepatitis B, AIDS or HIV is very good at this, and Epstein-Barr. So a lot of these viruses do this by incorporating their genetic material into the host, but then kind of shutting off or having mechanisms that shut off the transcription and translation of that viral DNA. And by doing this, they kind of just hang out in the cell in a resting state. This allows for any time that that cell multiplies normally or divides normally, it kind of expands the amount of the virus present in the body. And then some kind of response or some kind of stimuli occurs and it kind of wakes up the virus, and then it becomes infectious and starts doing its job. And then a sequelae is a long-term or permanent damage to tissues or organs. So generally, sequelae is kind of thought as a consequence of having a disease. So reservoirs, so this is the primary habitat in the natural world where pathogens originate. So being a good pathogen, you need to be able to survive in the environment as well, because that's where you're going to be taken into your host. You don't have to survive, you don't have to multiply extremely fast or happily, but you have to be able to survive. So there are living reservoirs. They're generally considered carriers. You have asymptomatic carriers. These are people who do not exhibit symptoms, but they still carry the organism. You have incubation carriers. These are where the organism kind of incubates, grows, and divides, but doesn't necessarily make the person sick, but they can then transmit it on. You have convalescent carriers. These are people who are getting over the disease. And as they're getting over it, they're still infectious. You have chronic carriers. These are people who are always carrying the organism but don't always explain or display symptoms. And then you have passive carriers. These are people who transiently carry the microbe. It's not actually growing and dividing in them, but they get it, they transmit it, and then it goes away. So here's a nice little example of all of these in a picture format. So animals as reservoirs. We have vectors. So this is a live animal that transmits an infectious agent from one host to another. A good example of this would be mosquitoes. A lot of these are insects that like to land on people. And there's two different types of vectors. You have a biological vector, so this is where part of the pathogen's life cycle is associated with the vector. So generally it's internal. Mosquitoes are a good example of this, tsetse flies, ticks. All of these guys have microbes that utilize the structure and the function of the tick or the ectoparasite or the mosquito and take advantage of that to transmit the organism. Mechanical vectors, on the other hand, just carry microorganisms that could be pathogens on their body, and then when they land on something or they brush up against something, it transmits the organism that way. It doesn't actually go internal, and it's not necessarily a part of the life cycle of the pathogen. So here you can see, here you can see a mechanical vector. So generally flies like to land on poop. It's where they lay eggs. They can carry around E. coli in their legs and then transmit it to subsequent people or subsequent animals or whatever the case may be. Mosquitoes, on the other hand, actually actively take up blood, and in that blood you can find things like malarial parasites, and those malarial parasites actually utilize the digestive tract of the mosquito to continue out its life cycle and transmit the malaria. And this thing doesn't want to advance to the next slide. Okay. So zoonosis. This is an infection that's typically found in animals, but has been somehow transmitted to humans. Whether that's through mutation in the pathogen or just the fact that we've come into close contact with animals we've never seen before. But this allows for kind of a transition of an animal pathogen into humans. Most really good pathogens that we see today were originally found in animals, and they are zoonoses. Things like HIV, influenza, hepatitis. These are things that were found in animals, SARS, and then they were transmitted to humans. And generally, it's happening quicker and quicker as we evolve. So if you look in like the 1900s compared to now, there's a lot more zoonoses now than there were then. And it has a lot to do with the fact that we're spreading ourselves out as a human population. So we're going into forests, we're going into areas that we haven't been to before, and we're becoming exposed to different types of animals that we've never seen before, in close contact with these animals. And that allows for these kind of pathogens that are associated with them to kind of transition into us. That's how HIV started, that's how SARS started. But it can be somewhat problematic. Excuse me. So here are a few common examples. For rabies, you can see, or viruses, you can see rabies, yellow fever, viral fever, hantavirus, influenza, West Nile virus, all these guys you've heard about before. For bacteria, things like plague, salmonellosis, anthrax, all found in animals first and then transmitted to us. And parasites are notorious for this. Almost every parasite has an animal reservoir of some sort. So non-living reservoirs, this can be the soil, it can be water, it can be an environment, it can just be anywhere the bacteria or pathogen needs to hang out before it can infect someone else. So how are things transmitted, or are these pathogens transmitted? You can have a communicable disease. This is the vast majority of them. It can be transmitted from one host to another host, whether that's through saliva in the mouth, through blood, through urine, through feces, whatever the case may be, it's still transmitted directly or indirectly from one human to another. Non-communicable diseases does not arise through transmission of the infectious host from a host, but it's acquired through some other circumstance. Whether that's becoming compromised, immunocompromised, and your normal flora actually become pathogenic, or you acquire it from a non-living reservoir and you don't really know where it came from. So maybe I got influenza from licking a doorknob. Well, I probably got it from someone who had influenza, but I can't make that connection directly because I'm not 100% sure, so it's a non-communicable disease or non-communicable route of transmission. So patterns of transmission, so this is pretty much common sense. You can see contact, droplets, vertical, so from mother to child. Biological vectors such as mosquitoes or ticks or whatever the case may be. Can also be from fomites, bacterial water. Fomites are non-living vectors. And then air is also obviously a way of transmitting organisms. So you can have contact transmission or indirect transmission. So indirect transmission uses some sort of vehicle of either transmitting the organism or allowing the organism to kind of hang out and wait for a good host. There can be things like fomites, again, which are inanimate objects that hold pathogens, food, water, biological products, be the air, whether it be aerosols from sneezing. So when you sneeze, you shoot out aerosols a good 20 feet, and then they can kind of float around. They're not heavy enough to fall to the ground, but they just stay lofted in the air. That's how things are transmitted. Or droplet nuclei. These are the larger water droplets, which actually fall to the ground fairly rapidly, but they carry a lot of organisms with them, so they do become fairly infectious. So direct contact, it's touching, kissing, sexual intercourse. Perinatally, so that's crossing through the placenta, or perinatally, that's during or shortly after birth. So HIV is transmitted perinatally, but toxoplasmosis is transmitted prenatally. There's this nice little, there's the droplet nuclei we talked about, and then you have these going far out this direction. You have all of these aerosolized particles that contain infectious agents. You can see this guy's from 1940. It's a very famous photograph. You'll see it in almost every microbiology textbook. Lastly, what are nosocomial infections? So these nosocomial infections are infections that occur in a hospital setting. Typically, these can be these infections tend to target people who are immunocompromised, and whether that's because hospitals are full of immunocompromised people or the pathogens have evolved into affecting immunocompromised people is yet to be determined. But because you're putting a lot of people, a lot of sick people in one area, and if you're a hospital that isn't very good at warding off infected patients, you can run into a lot of problems. So if you have immunocompromised ward in one portion of the hospital, then right next door you have the Infectious Disease Center. You can see how that can be somewhat problematic. You're going to basically make people who are very susceptible to getting pathogenic infections come close to pathogens on a fairly regular basis, and you'll run into problems. So there's a lot of good hospital practices that have come about to kind of make sure that these things don't happen as often. But nosocomial infections are a breeding ground for evolution in microorganisms, especially when it comes to resistance to both chemicals and antibiotics. And that is because they're under constant natural selection pressure. Constant pressure of using either the chemicals to disinfect everything because hospitals are notoriously clean, or you're surrounded by patients who are on multiple cocktails of antibiotics. So it's kind of a nice breeding ground for these resistant microbes, and for that reason you get some nasty nosocomial infections such as MRSA, which is methicillin-resistant Staphylococcus aureus, which is almost resistant to almost all the antibiotics that we currently use, and it likes to infect skin and cause necrosis or dead skin, open oozing sores, which is pretty disgusting. So there are about 2 to 4 million cases per year, and it's important to follow medical asepsis, so sterilizing, keeping things clean, and making sure that if you are using a chemical to disinfect, that you use it to its full extent and you disinfect everything, don't leave anything behind. You can see the vast majority of urinary tract infections, and that's because a lot of people get catheters, and catheters tend to get biofilms around them, and it just is a breeding ground for pathogens. But you can see respiratory, surgical sites, meningitises, gastrointestinal, skin, septicemia. You get a little flavor of everything. So, universal blood and bodily fluid precautions, or universal precautions. So this is a guideline from the CDC on a way to prevent illness spread from blood and other bodily fluids. So the biggest thing from this is to assume that all patient specimens are infectious. Just because you know someone or you know that they're not infectious, don't treat their blood any differently than someone else. Just treat it all the same, and then you have nothing to worry about. So what causes a disease? It's called the etiological agent. That is the organism that has caused the disease you see in a patient. And sometimes it's easy, sometimes it's hard to determine. When it's hard to determine, that's when it becomes difficult to treat the patient because you don't know exactly what to target. So the first development in determining the etiological agent is Cox Postulates, which we've talked about before. And this kind of developed a standardized method, but the problem with this method is that it uses phenotypic analysis, and it also requires the organism to be culturable, and not all organisms are culturable. So we've talked about this before, so find evidence of a particular microbe in every case of the disease, isolate that microbe from an infected subject and cultivate it in pure culture in the laboratory, inoculate a susceptible healthy subject with the laboratory isolate and observe the same resultant disease, and then re-isolate the agent from the subject. So it's a long process, and again, it requires the ability to culture the organism. Also, there are some ethical issues here, so if it's a disease that's only capable of infecting humans, like HIV, do you re-inoculate a healthy human with your causative agent to see if it is HIV? Probably not. So there's ways around Cox postulates, such as doing genotypic analysis, so sequencing, or maybe doing PCR, looking at the actual DNA, the genes that are associated with the organism, make it much, much easier, much, much quicker, and less ethically problematic. So here's a nice little picture if you're interested. So quickly about epidemiology, I'm not a huge fan of epidemiology, but I do need to talk about it a little bit. What epidemiology is the tracking of diseases geographically. So looking at where a disease starts, maybe in a country or a city or wherever, and then watching it spread and trying to determine what's allowing it to spread and how to prevent it. So epidemiology is the study of occurrences of diseases. So an epidemic is a sudden acute disease outbreak affecting many people. So it's within a localized area. A pandemic is a worldwide epidemic. And then an endemic is a chronic occurrence in a geographical region. So epidemics are sudden and they're acute. So it could be a thing like a foodborne illness or something from eating infected vegetables in that area. A pandemic would be if those vegetables were transmitted throughout the world and everyone got sick with that same pathogen throughout the world, no boundaries. And then an endemic is something like a cold that happens every single year. It's not sporadic, you know it's going to happen, and it's within a specific centralized location. And then sporadic are just kind of random outbreaks, and these can be the hardest to determine because you're trying to figure out why one's happening here, why one's happening here, why one's happening here, and sometimes there's no real connection. So here's an endemic occurrence. So it happens fairly regularly in these known areas. We know it's going to happen. Here's epidemics. So they're sporadic, focal in their location, but they can be spread out. Sporadic, small cases, kind of all over, no real pattern. And then a pandemic is just global. And this is because of travel. The ability to travel around the world within 24 hours makes it very easy for things to spread. Okay, come on. Next. Okay. So who, when, and where the tracking disease in the population. So epidemiologists are concerned with virulence, portal of entry, portal of exit, and the course of a disease. And they are interested in surveillance. So there's this whole kind of system set up for reportable diseases. So by law, there are a handful of diseases that must be reported to the CDC every single case that is received. And that allows the CDC to kind of track where these diseases are migrating, where they're coming from, and ways to prevent that. So methods of epidemiology, so you can get sources of information either from public record questionnaires, surveys, hospital records. There's a little bit of this information almost everywhere. It's just how you utilize it, how you put your resources together to fully extract it. You also have notifiable diseases, so these are diseases that are required by the CDC for doctors to let the CDC know that they have a case of this disease. And they're generally things that are somewhat serious, also can transmit or be transmitted fairly easily, like HIV is a notifiable disease. Now the doctor doesn't have to say, okay, so-and-so has HIV. They don't have to give any of the patient information. They just have to say, I live in this community and this person has HIV, and you could track it. And then they look at the statistics of this. So you look at the incidence rate or the prevalence rate. So the incidence rate is how many cases you have in a specific area at any given time. The prevalence rate is how that is going to change over time. So how much more you have from point A to point B, or how much less you have from point A to point B. Where the incidence rate is how much you have at any given time. It's like a snapshot. Here are the reportable diseases in the United States. So you can see things that are spread fairly easily, like gonorrhea are included. But you also see things that are pretty nasty and that we don't really see too often, but when we do, you need to report it, like rabies, for instance, or plague. So how do you measure infection? So there are some experimental procedures with animal models, but again, this can be problematic because you're looking at an animal model against a human model, how similar, how different they are, and all that good stuff. But sometimes it's the only thing you have. So you're looking at the infectious dose or the morbidity. So how much it takes for the animal to become sick. So morbidity is kind of the yicky feeling, being sick, not being able to work, not being able to do this or that, but not necessarily death. The ID50, so this is the number of cells required to infect 50% of the subjects. And then there's the lethal dose, or the mortality, the LD50. So this is the number of cells required to cause death in 50% of the subjects. So morbidity is just icky feeling, sickness, general blah, where mortality is death. And we're done. So enjoy.